The organic substances that we need in small amounts in our diet and whose deficiency leads to specific diseases are called vitamins. When vitamins were first identified, they were found to have amino groups. Thus, the word vitamin originated from vital plus amine. However, nowadays, most known vitamins don't necessarily contain amino groups. Hence, the word vitamin has been replaced by vitamin. Vitamins perform specific biological functions. They are essential for proper growth and to carry out many vital functions of the body. Most vitamins are involved in utilizing the major nutrients like proteins, fats, and carbohydrates. That is why a diet deficient in vitamins leads to specific diseases. Note that high doses of vitamins can also lead to health problems. Check with your doctor before taking vitamins. When vitamins were first discovered, their chemical nature was not completely known. Therefore, they were named after the alphabets A, B, C, D, and so on. Later, the chemical nature of vitamins was established, and therefore, now they are referred to by their chemical names, such as retinol, thiamine, riboflavin, ascorbic acid, cholecalciferol, and so on. However, the vitamins are now referred to by their alphabetical nomenclature also. Let us now look at the classification of vitamins on the basis of their solubility. Vitamins are typically classified into two groups based on their solubility in fat or water-fat soluble vitamins and water-soluble vitamins. Fat-soluble vitamins are soluble in fats or oils. They are stored in the liver and adipose or fat-storing tissues. Vitamins A, D, E, and K are fat-soluble vitamins. On the other hand, water-soluble vitamins are soluble in water. They are readily excreted and therefore cannot be stored or accumulated in the body. They must be a regular part of your diet. B group vitamins and vitamin C are water-soluble vitamins. Let us study some important aspects of vitamins with respect to their sources and the diseases that their deficiency cause. Vitamin A is an important fat-soluble vitamin. The deficiency of vitamin A leads to xerophthalmia, that is, night blindness, in which the central portion of the eye, the cornea, becomes opaque and soft. Images of xerophthalmia are quite disturbing. Vitamin D is another important fat-soluble vitamin. Your skin produces vitamin D when exposed to sunlight. Food sources of vitamin D include fish and egg yolk. This vitamin is required for bone growth and calcium metabolism. Vitamin D deficiency leads to rickets, that is, bone deformities in children, including soft and weaker bones. In adults, this is called osteomalacia, characterized by soft bones and joint pains. Another important fat-soluble vitamin is vitamin E. Dietary sources of vitamin E include vegetable oils and cereal grains. Vitamin E deficiency leads to more fragile red blood cells and muscular weakness. Vitamin K is another important fat-soluble vitamin. Green leafy vegetables are good dietary sources of vitamin K. Vitamin K deficiency leads to slower blood clotting. Vitamin C, or ascorbic acid, is an important water-soluble vitamin. Dietary sources of vitamin C include citrus fruits, amla, and green leafy vegetables. This vitamin is required for the synthesis of collagen and for the calcification of the bones and the teeth. Vitamin C deficiency causes scurvy, characterized by bleeding gums, deformity in bones, etc. B group vitamins are another group of water-soluble vitamins. They are found in meat products, whole grains, milk, and yeast. They are essential for metabolism and the proper utilization of energy sources like carbohydrates, proteins, and fats. B group vitamins include vitamin B1, vitamin B2, vitamin B6, and vitamin B12. Vitamin B1, or thiamine, was the first vitamin to be discovered. The richest source of this vitamin is yeast and the outer layers of cereals like rice, wheat, and millets. A deficiency of this vitamin causes beriberi, which is associated with loss of appetite and retarded growth. Vitamin B2, or riboflavin, is mostly found in milk and milk products, eggs, liver, green leafy vegetables, pulses, wheat, and so on. Insufficient vitamin B2 leads to chelosis, which is characterized by fissuring at the corners of the mouth and lips, digestive problems, and burning sensation in the skin. Vitamin B6, or pyridoxine, is found in meat, liver, egg yolk, cereals, grains, and vegetables. Its deficiency causes convulsions. Vitamin B12, or cobalamin, is found in meat, fish, egg, and curd. Its deficiency leads to pernicious anemia, that is, RBC deficient in hemoglobin. Nucleic acids are biomolecules present in all living cells and are responsible for transmitting genetic information from one generation to the next. In other words, they are the carriers of heredity. They are found to exist along with certain proteins. Proteins, along with the nucleic acids, constitute nucleoproteins. Now, in this context, let us look at the chemical composition and structure of nucleic acids. The The basic components of nucleic acids include a pentose sugar, a phosphate group, and nitrogen-containing heterocyclic compounds called nitrogen bases. Depending on the type of the pentose sugar present, nucleic acids can be broadly classified into DNA deoxyribonucleic acid and RNA ribonucleic acid. Examine structures 1 and 2 given here. From the structures, we can say that in DNA, the sugar is beta-D2-deoxyribose, whereas in RNA, the sugar is beta-D-ribose. Let us now look at how these two nucleic acids differ with respect to nitrogen bases. There are 4 nitrogenous bases present in DNA: adenine, guanine, cytosine, and thymine. There are also 4 bases in RNA. In RNA, the base uracil is substituted for thymine. Note the structural similarities between adenine and guanine and between cytosine, thymine, and uracil. On the basis of the structural similarities among the nitrogen bases, they are broadly categorized into purines and pyrimidines. Adenine and guanine are purines, while cytosine, thymine, and uracil are pyrimidines. A nitrogenous base attached to the C1 prime carbon of the sugar is called a nucleoside or a base sugar unit. The nucleoside, adenosine, is shown here. Note that we number the sugar carbons in nucleosides as 1', 2', and so on as shown. A pentose sugar with the nitrogen base at the C1 prime carbon and a phosphate group at the C5 prime position is called a nucleotide, a base sugar phosphate unit. Look at the structure of a nucleotide of DNA and RNA. You can see that there is only one hydroxyl group attached to the sugar ring of DNA, while there are 2 hydroxyl groups attached to the sugar ring of RNA. Try to answer this question on your own before checking the answer. What products would be formed when a nucleotide from RNA, which contains cytosine, is hydrolyzed? Start with the correct nucleotide structure. Hydrolysis of this nucleotide would produce phosphoric acid, beta-D-ribose, and cytosine. Nucleotides are linked together by phosphodiester linkages. These nucleotides, which are linked by phosphate ester bonds, connect the 5' phosphate group to the hydroxyl group of the sugar at the 3' position, as shown in the diagram. The backbone of a polynucleotide chain is a repeating sequence of sugar phosphate. The sequence of nucleotides in the chain of a nucleic acid is the primary structure of the nucleic acid DNA or RNA. The sequence in which the 4 nitrogen bases are attached to the sugar phosphate backbone of a DNA strand is called the primary structure of DNA. DNA forms a secondary structure in the form of a double-stranded helix, like a twisted ladder. Two strands of DNA interact to form the double helix, where the sugar phosphate backbone form the rails of the ladder, while pairs of nitrogenous bases, either purines or pyrimidines, form the rungs or steps of the ladder. James Watson and Francis Crick determined the double helix nature of DNA using X-ray crystallographic data collected by Rosalind Franklin. The two strands of DNA are held together by hydrogen bonds between specific pairs of bases. Adenine always pairs with thymine. Cytosine always pairs with guanine. As the two strands are held together by hydrogen bonds between specific pairs of bases, the strands are said to be complementary to each other. Notice that the complementary strands run in opposite directions. Let's look at the nucleotide pairing in greater detail. A purine base of one strand is paired with a pyrimidine base on the other strand. Two hydrogen bonds are formed when adenine pairs up with thymine. When a guanine base pairs with cytosine, three hydrogen bonds form between the bases. Try to answer this question on your own before checking the answer. In RNA, which nitrogenous bases form hydrogen bonds with uracil? The structure of uracil is shown here. Notice the structural similarities between uracil and thymine. Therefore, in RNA, uracil forms a complementary base pair with adenine. Two hydrogen bonds are present between the bases. Can you answer this question on your own? Given here is the sequence of nucleotides in a strand of DNA. What is the nucleotide sequence in the complementary strand? Remember that the complementary strand runs in the opposite direction and that T pairs with A, while C pairs with G. Therefore, the sequence of the complementary strand can be readily worked out. While DNA is found as a double helix, RNA is single-stranded. The strand may fold back on itself to form helical motifs, such as stem loops or hairpin structures. There are three important types of RNA in living systems. Messenger RNA carries genetic information from DNA to ribosomes for the synthesis of proteins. Transfer RNA brings amino acids to the messenger RNA. Ribosomal RNA is the RNA component of the ribosome where proteins are synthesized in the cell. Nucleic acids play a number of important biological roles. They are the chemical basis of heredity, storing genetic information while also allowing for changes between generations. Each species has a unique genetic code with a specific number of chromosomes. DNA copies itself or self-replicates during cell division, so that identical copies of the genetic code are transferred to the daughter cells. Nucleic acids are vital for protein synthesis. DNA stores the code for the amino acid sequence, while RNA molecules link the amino acids together. Each individual has a unique DNA sequence. Information regarding the sequence of bases on DNA is called DNA fingerprinting. Through DNA fingerprinting, it is possible to determine if two samples of DNA belong to the same person, two related individuals, or to unrelated people. DNA fingerprinting has important uses in identifying criminals, in determining paternity, identifying accident victims, and in the study of evolutionary changes. Based on the data shown, did any of the suspects leave DNA behind at the crime scene? Based on the DNA fingerprinting, we can eliminate suspects 1 and 3 as their patterns do not match the DNA found at the crime scene. It seems likely, however, that suspect 2 may have been at the crime scene.